So it's my pleasure today to tell you a little bit about my story. PJ asked me to kind of cover my career, my science, and also give you tips on getting into graduate school. So those are three sort of diverse things. So we'll see how far we get. I did make some handouts on the tips for getting into graduate school. So if we don't get through all of those, maybe you can take those with you. I actually started out in the middle of the country. I'm a native of Kansas, in case anybody doesn't know what state that is. I actually grew up on a dairy farm outside a very small town. And it was the place where I learned to work. And that's a very good criteria or skill to have as if you want to be a graduate school, want to go to graduate school, because it is hard work. So I went from this little tiny town to Lawrence, Kansas, where the University of Kansas is located. There I had the opportunity, I was very fortunate as a high school student, to actually go for a couple summers and do research at the University of Kansas. So I was pretty sure when I was going to go to college that that's where I wanted to go, because I already had a lot of connections and actually a job waiting for me. So I had, and that was really a very enriching experience, was the summer the 2 summers I spent there before actually going on to college, and was a good introduction for somebody who was literally fresh off the farm. So I worked in microbiology. I worked in 2 rather different fields, which I must say have followed the rest of my life. The first was I started out in radiation biology. They actually had a nuclear reactor at the University of Kansas back then. And so they irradiated things, and I worked on this little tiny plant called Haplopappus gracilis, which was very useful for looking at chromosome aberrations, because as you can see from this picture, it actually had only 2 sets of chromosomes, so it was very easy to tell if something had gone wrong with those. So I started out my science career looking at things through the microscope, and I still do that today. So the first 2 years I was at Kansas, I worked there in that laboratory in that region. And then I took a course in immunology from a really fantastic professor. And from that day forth, I knew I wanted to be an immunologist. So I sort of never looked back, if you will. And actually, I started doing work on lymphocytes at that time, actually ones from the rabbit appendix. Did you know that the appendix is a lymphoid organ? It's actually just composed of lymphocytes, although the function of it is a little bit unclear. I got my degree in microbiology in 1970, and then I was very fortunate to receive an award called the Woodrow Wilson Award, which guaranteed a graduate stipend to whatever graduate school I wanted to go to. So I was actually, I was the only one awarded in microbiology in 1970. They do still give these awards. It's still a very prestigious foundation. So I decided, after looking at a lot of schools, that I wanted to go west. And so I traveled across the country to University of California at San Diego. This is the University of California today. It looked a bit different when I was there. So you can see it's now a very large campus. It's bordered by the Pacific Ocean. At that time, it was really the beginning of immunology in many ways, but it was a very fantastic place to be, because there was not only the university, there was the Scripps Institute, there's a Scripps Institute of Oceanography, but there's also a Biomedical Institute, and there was the Salk Institute that was founded by Jonas Salk, all within like a mile of each other. And I worked with a professor who had moved there from Scripps up to the university, And he had worked together with another professor and was the first to actually culture lymphocytes. So these are the immune cells that are the most important for adaptive immunity. And they were very interested in being able to look at these cells to identify responses of B cells to different antigens. And we used something called the Urney plaque assay, which again was new at the time, that you could actually see individual antibody-producing cells and count them. Again, a breakthrough at that point. Now it's a really old hat, but and they were also very interested in the other kind of lymphocyte called T lymphocytes. And I'm going to talk a lot more about those. They actually help B cells make antibody and then also do a lot of other things. And I was on a project that, and I will be perfectly honest and say I stumbled a lot as a graduate student, a lot of false starts, a lot of things that didn't work. But again, the key to graduate school, I think, is perseverance. And fortunately, I had some good mentors that helped me with that. And actually, in that process, so these are the B cells that most people think about. They make a lot of antibody of different types. They require T cells to help them do this. But I actually discovered and identified another totally different kind of B cells that are called B1 versus the conventional B2 cells. They make only a certain type of immunoglobulin, and they're really autoreactive cells. So they react with self-proteins more than anything else, and they're found in sort of unique places in the body. I was able to publish 5 papers over the course of my graduate career and got my PhD in 1975. Now, like many other young people at the time, I had a boyfriend, and so when the next step came, it was a question, what do I do? Do I go where he is? Do I go someplace else? Again, I looked at a lot of places. My heart won at that point, and I traveled up the coast to San Francisco. So went from one big city to another. And went to the University of California at San Francisco. And actually worked in this building here, which these very tall buildings would actually sway about 5 or 6 inches during earthquakes. So it was exciting if you were on one of the top floors. But it was a great place to be. A lot of things were going on at that time. And interestingly, I worked on a sort of unusual thing looking at the mentor I worked with had identified that there was an immunosuppressive factor in human seminal plasma, and I did quite a bit of work with that. He wasn't a great mentor, but I collaborated with somebody at the University of California, Berkeley, who was very good, and as a result, I managed to get 3 papers out. Now, I was actually only there a year and a half because, again, my boyfriend had a job offer at the University of Rochester, which I thought was the end of the earth, quite honestly. I'd never heard of this place, right? It seemed like it was a long ways away, but they made me a good offer as well, and so I went across the country to Rochester because I thought it would be an interesting place to spend 4 or 5 years. So here I am many years later, actually, having not escaped from Rochester. So have become an East Coaster, although the real people on the East Coast say Rochester is not the East Coast, right? But if you're from California, it is, right? And at the time I came, the University of Rochester Cancer Center had just been formed. I was actually recruited into the Cancer Center. And so this was when I started working on tumor immunology. All of my previous training was not in cancer, not in tumors, but I had been an immunologist for most of my training. And we were very interested in what was happening in the tumor microenvironment. So, and again, I brought back things from my undergraduate days. I was actually hired by a radiation biologist who was interested in how the immune system affected the response to radiation of tumors. And so I started a study using these are balls of tumor cells that were grown in vitro. They were a good model because they receive oxygen only from the outside, so that the inner cells, here you can tell this is a cross-section of one of these, so if you just cut one of these in half, it would look like this. And these cells in the center are actually dead because they haven't received enough oxygen, which is what also happens in tumors at a distance from the vasculature. So we continued work doing this and a lot of other things. So I want to just digress for a moment. So I became very interested in T cells because it was recognized even then that they were very important in terms of the immune cells that could actually kill tumor cells. And they do this directly by direct interaction as shown. So these are a couple tumor cells that are being attacked by these T cells. And these are a particular kind of T cells. There are a lot of different kinds of T cells. These happen to have a marker on them called CD8. So we can detect them using that marker, and it's very effective. Now, one of the things that was clear to me was because you have to have this direct interaction between the tumor cells and the T cells, that it seemed to me the interesting place to look at these was in the tumor. Now, everybody else was looking at immune cells in the spleen and the lymph nodes and where immune cells are usually found, but it was clear to me that the action was happening within the tumor. And interestingly, the radiation biologists didn't know anything about immunology, and basically they wanted to use tumors that had no immune cells in them. They weren't interested in their experiments being contaminated by immune cells. But the more I looked, the more I saw that all tumors had a lot of immune cells in them and clearly were playing a factor and a role. So one of the things I quickly found out is you have to think about how these cells get into the tumors, and obviously that's through the blood vessels. So on the next slide, so I had to, because it was sort of part of it, I had to get interested in how blood vessels form in tumors. So tumor cells, even though they start out small and will use vessels that are in the tissue where they arise, they grow very rapidly, and they need more blood vessels to provide them, particularly with oxygen. That's the most limiting nutrient, but also all of the other things that they need to grow. And so they respond to this lack of oxygen by making angiogenic factors. And so they cause the blood vessels to sprout, and you get new vessels formed. The only problem is these vessels are not normal. So generally, angiogenesis, when it occurs, when you have new blood vessels form, there is a balance between pro-angiogenic factors and anti-angiogenic factors. So it's a very controlled process. But with the tumors, when they're pumping out all of these angiogenic factors, it becomes abnormal. And so you end up with vessels that are very large, and not very well constructed. And so they're actually not very efficient at providing oxygen to the tumors or other things, right? This is some images I took from a paper in the literature. On the top are blood vessels. So these are EM micrographs of vessels in different kinds of tissues. So this is colon, this is the skin, and this is the skeleton, because the blood vessels run along the muscle fibers, so they're these very long, straight things. And then these are similar vessels that are in tumors in those organs. So you can see they're vastly different, right? These, they become very convoluted, lots of they end sometimes in just dead ends. And they're usually larger and less efficient, because the larger something is, the less the blood flow. So the next slide. And I don't know whether we can have some lights down. I have some pretty pictures, but you can't see them in. Yes, that's good, right. So these are pictures that were actually taken of a mouse tumor growing in, that was done in my laboratory. Can you see okay? I'm sorry, we can put it up and down. So this is where a tumor was actually implanted in the leg muscle of a mouse. So these are the vessels, as I saw on the previous slide, that are part of the normal muscle. The tumor is growing over here. It is making factors that is causing this increase in angiogenesis in this kind of dividing line between the normal vessels. And you can see these tumor vessels are really very chaotic and turn out not to be very functional. So next slide. So that is important to think about this for several reasons. So when we treat tumors, it's very important that there is good vascularization to the tumors because everything is impacted by that. So if we treat tumors with chemotherapy, which is one of the major modalities, those treatments are given intravenously. And so what happens, as shown on this little diagram where this is a blood capillary, if the drug is here and it diffuses out from this, the cells that are closest to the blood vessel will utilize the drug, and the further you get from the vessel, the less active drug there is. So the farther away from the blood vessels, these cells become resistant simply because they don't receive as much drug. The same problem occurs for radiation therapy. So even though you irradiate the whole tumor, the levels of oxygen are very important because it's the formation of oxygen-free radicals that actually damage the DNA and why radiation causes the damage that it does. So without oxygen or with lower levels of oxygen, there's less damage. So the question I had was, how do these alterations in oxygen affect immunotherapy? Because again, remember the same thing. If we're going to have cells that are going to kill tumor cells, they have to get in through the blood vessels. So the question was, how effective are they at moving away from these blood vessels and actually functioning in areas where there's very little oxygen? So at this time, which is sort of the late 1980s, there was no way to measure hypoxia. There was a lot of interest from the radiation biologists because they realized the importance of this, and they realized that some tumors responded very much better to radiation than did others. There was a lot of heterogeneity. And so there were drugs that had been developed to try to sensitize tumors to radiation. And this was, and they really acted by trying to bind specifically to these hypoxic cells. But there were problems with most of those, and even so, if you put drug in, how were you going to, quote, see that drug, see what cells had taken it up? Well, as an immunologist, there's an easy answer to that. You make an antibody against it, and you tag that antibody with something that you can see. Now, the problem is all this work was being done by radiation biologists, and they knew no immunology and were pretty hopeless at it, actually. But interestingly, so I had a colleague who was working on this question at the University of Pennsylvania, and it turned out he had been a postdoc at University of California, San Francisco at the same time I was, and we actually carpooled into work together because it was a long drive across the Golden Gate Bridge to get to work. And so we teamed up, and he developed a new drug, which is shown on the next slide. Which he called EF5. This is the structure of it. So you can see it's a very small molecule. And these are very difficult things to make antibodies against, so you have to couple them to something else. So we coupled it to a protein because he was a good chemist. I didn't know any chemistry. So it turns out that we were able to make antibodies against this, and this was the time when we were just learning how to make what are called monoclonal antibodies. So we could actually, and this is a very clever technique where you take a cell that's making an antibody, you fuse it with an immortal cell, and you can actually get a cell line that produces a single antibody. So they're called monoclonal because there's only one type. And for the first time, you could make buckets of this, right? And you could have all the antibody you wanted, and it was very highly specific. So we had a really good system to be able to detect hypoxic cells, and therefore we could all go about answering the questions that we wanted to. So these are actually some recent pictures that a student took. And this is, again, one of those mouse tumors that I told you about that has been stained for a marker that is expressed on blood vessels. So these purple things that you see here are actually small parts of blood vessels. She also stained them for cells, the T cells. These are, you can't see this very well, but CD8. So you can see these are the T cells that are capable of killing tumor cells. And the question we had was, where are these relative to both blood vessels? And I think you can see that these are primarily near the blood vessels. There's areas here which don't have much blood vessels, and there's fewer of these cells. And we also stained them with this marker for hypoxia, which is shown on the next. So if you overlay this, so the red are areas that are very low in oxygen. And again, you can see those are areas where there aren't very many blood vessels, and there are many fewer of these CD8 cells here. So this tells us that these hypoxic cells are also probably going to be more resistant to our killer T cells because, again, those cells have to get and be able to actually contact the tumor cells for this to work. So this illustrates just one of the problems in trying to think about immunotherapy for tumors, and there are others as well, although some of those are getting solved. This one has not quite been solved yet. But. So just to tell you a little bit more about anti-tumor immunity, because I'm not sure how familiar everybody is, but the immune cells, of course, are specific. They recognize antigens that are different from self. And that's one of the problems with having immunity against tumors, is tumors are a lot more like ourselves than they are something foreign. A virus or a bacteria is very different from us, right? Now, a tumor cell is different. Otherwise, it wouldn't be a tumor cell, right? And most of them have some mutations, or they have other things that are recognizable by the immune system. So one of the things that is being worked on a lot is trying to figure out what those antigens are and trying to stimulate better responses against those. So we need to identify what those antigens are. We then need to be able to increase the number of cells that are capable of reacting with those and being able to improve their function. Now, there are a lot of different kinds of immune cells. I won't go into all of that because even I don't understand it all. But one of the things we do know is that immune cells communicate via small proteins called cytokines. And so these are very important molecules that allow cells to talk to each other, that stimulate them to do one thing or turn them off. So they're very important molecules. And we've done a lot of work looking at these cytokines and their role in stimulating tumor immunity. And some of the things that we've looked at are IL-2, IL-12, and also interferon gamma. Now, IL-12 was originally known as T-cell growth factor, and it was really the discovery of this that allowed people to be able to culture T cells and expand those. So these have been a very key thing. IL-12, it turns out, is also very stimulatory to the T cells and also affects the blood vessels, which is one of the things that we've worked on. So this is just a slide that illustrates how the presence of larger amounts of these cytokines can alter the vessels within tumors. So this is a tumor that's called B16. It's a melanoma, a skin tumor that people get when they're out in the sun too long. This happens to be a mouse tumor. And you can see these vessels are these very large chaotic vessels, as we've seen in previous slides. However, we've taken these cell lines, it's a cell line. We can modify it by putting in genes for either IL-2 or IL-12, and that modifies what these look like. So the IL-2 tumors, actually, their vessels look a bit more normal. They're smaller, cover more of the area. And the same is true for IL-12. In fact, these have the most normal vessels. And there are several factors. Interferon gamma is involved in changing this vasculature. These cytokines, they affect the tumor cells directly, or I'm sorry, the immune cells, but they also affect the vessels. So they can have a sort of at least two-pronged effect by altering how the T cells can access the tumors, but then also how they function there. So the next slide. So, and this illustrates how they can affect the infiltration of T cells. So two things are shown on this. So these top ones are pictures of the blood vessels. So that's shown in the green here. And the second ones have been stained for these CD8 cytotoxic T cells. So there's very few. There are some in the B16 tumor. But when you add this IL-2 growth factor, you can see now that there are a lot more of these, and they have a very interesting pattern. So they mirror the blood vessels. So these are very close to the vessels, within a few cell diameters usually. So they don't get out into the rest of the tumor very well. So these tumors grow a bit differently than these, but still they grow out. The IL-12 also have an increased number of these CD8 cells, but they're more evenly dispersed, probably because of the difference in the blood vessels and probably some other differences in the tumor stroma and other factors as well, which still remains to be determined. So this is a good example of how these cytokines can affect the immune cells. There's more of these, but also affect the vessels. So just to go a little bit more in talking about, so I've been very interested in the immune cells, but obviously another thing, immunotherapy is still not the only thing for tumors, and almost all tumors are treated with some sort of conventional therapy. And again, because of my roots in studying radiation biology, I've continued to interact with radiation biologists, and I've really focused a lot of my work on how radiation affects the immune response and how the immune response affects the response to radiation. And as I've alluded to, I use these mouse tumor models. This is typical of these little black mice that we use. A common strain, because there have been a lot of different modifications of this strain made with different molecules that are either knocked in or knocked out, so they're a rich strain to use. And we implant these with a tumor that originated from one of these mice a while ago, and that is actually arose in the colon. So we inject these actually in the leg. We let those grow. By that time, they'll become well vascularized, as you saw in one of the earlier slides. And then at day 7, we give these a single dose of radiation. And then we look at how those tumors grow and other immune factors after that. So this is just an illustration of how these tumors grow. So what we're measuring is how big that tumor gets in the leg by measuring the diameter. And this is over time. So this is one where the tumors were just implanted in several mice, and this is the mean growth curve. So you can see these tumors grow very aggressively, right? These mice by about day 15, these are tumors that we would not let get bigger than that, that we would sacrifice the mice. But if they were irradiated back here at day 7, you can see that they continue to grow for a little bit, but then that growth is arrested. Unfortunately, these tumors over time will continue to grow out, so we haven't cured these mice. But we've looked a lot at what is happening in these tumors, and on the next slide, you can see that one of the things that's very important, so we think about the radiation being killing tumor cells, which it undoubtedly does, but what we've shown is that, and what's illustrated in this slide, is that without T cells, that radiation is virtually ineffective. So just to orient you to this, so these top two things are untreated tumors, and as we saw in earlier slides, these grow quite progressively. And this is what happens after they've been irradiated. Just like we saw in the other slide, they go down, they again will come up over time. But if you have treated the animals with an antibody against that marker on the cytotoxic T cells, the CD8, so that you've eliminated all of those, there's essentially no effect of the radiation. So even though these have been irradiated, in the absence of the T cells, these continue to grow. So we have shown this, now other investigators have shown this as well, and the radiation biologists now have to accept the fact that the immune response is pretty important for the response to radiation. People are now beginning, after many years, actually believing this. To show you that an important part of what those CD8 cells are doing is making a cytokine called interferon gamma that I talked about earlier. So we did a very similar experiment, except now we grow these mice either in or grow the tumors in regular mice, or we grow them in mice where the gene for interferon gamma has been knocked out. So these have T cells, but they can't not make this particular cytokine. And what we see under those circumstances is that, again, this is the effect we saw before, the progressive growth, the effect of radiation, where these go down. But if you don't have interferon gamma there, even though these have received exactly the same radiation, they still grow progressively. So particularly for this tumor model and for many other tumor models and in patients, T cells are essential for this response to radiation, and interferon gamma cytokine plays a very important role. It turns out it's a little bit difficult to treat patients with interferon gamma. So another cytokine has been used known as IL-12. And it is actually far more effective. So this is sort of the next steps of combining therapies. So now we're treating mice not only with radiation, but also we are treating them with this cytokine IL-12, which interestingly, one of its effects is it causes the production of a lot of interferon gamma. So a lot of this is probably an indirect effect. So again, this is what happens. These tumors grow very progressively. If you irradiate them, which is shown here in this dotted line, again, they stop growth and it levels off, but they will come back. If we have tumors treated with IL-12, they grow slower, but they still will progressively grow. But if we combine both of those things, so these tumors were irradiated and were also treated with IL-12, and in fact, these tumors not only go down, they stay down, and we followed these out literally hundreds of days, and these mice are cured. So I think this is a good example of what is probably needed today is combining therapies, because there are a lot of ways that tumor cells can evade the immune response or other therapies, and combining them hits them more than once and in different ways. So one of the newest therapies that has been developed recently and was actually approved, the first part of this was approved, I think, in 2011. So as I mentioned earlier, we have these T cells that we want to be able to kill tumor cells. They have to be stimulated in order to get them activated for that to happen. This is actually done by antigen being presented to them on a particular molecule. So that provides what is called signal 1. So T cells need at least 2 signals for stimulation. The second is provided by something called these co-stimulatory molecules, in this case CD28, that interacts with its ligand on the antigen-presenting cell. And that causes a positive signal, which then can allow that T cell to become an effector cell and kill tumor cells. But the immune response is a dangerous thing. So you want to be able to respond to things, but you also want to be able to turn off that response, because you don't want the response just going on and on after the antigen is depleted, because there can be collateral damage. It can cause a lot of damage to other things as well. And so there are a lot of built-in mechanisms to turn off the immune response. And so there are these other molecules. One of them is called CTLA-4, which is put on T cells over time as these cells become more differentiated. And it actually is a negative signal, so it turns the cell off, and so it does not get stimulated. There are also later signals that happen when that are effective when these become effector cells and are actually in the process of killing. And these are markers called PD-1, and its ligand is PD ligand 1. It is expressed on antigen-presenting cells, this ligand. Interestingly, it's also expressed on tumor cells. So by tumor cells expressing this, they can basically turn off this T cell and prevent it from killing. So these new therapies that have been developed actually block these things. And again, it's done with antibodies. So they've produced antibodies against this CTLA-4 marker, and so they can stop it from interacting here and stop the negative signal to the T cell. They've also made antibodies against this PD-1, which can prevent the ligand on the tumor cell from turning off the T cell. So these have turned out to be amazingly effective in humans. So now there have been trials done particularly in melanoma, because it's one of the most immunogenic, but now in other solid tumors as well. These have been FDA approved for melanoma, and now are being done in other cells as well. So very effective new therapies. I have just touched on a little bit of immunotherapy. If you're interested in this and want to do more reading, there was a great issue of Science came out earlier this month, actually April 3rd, which has a whole series of articles on cancer immunology. So if you want to do any more reading on this, very up-to-date, very readable articles that you might want to go to. I'd like to end this part by just acknowledging this is sort of my current lab group. I'd like to particularly acknowledge Dr. Scott Gerber, who was a graduate student with me, who went off and did a postdoc at Yale. I convinced him to come back. He worked in my lab. I helped run my lab for a while. He's just recently received a tenure-track position of his own. So I'm very happy that he's been launched, if you will, and we're going to be able to continue to work together. The other people here are all graduate students. Actually, this is Joanne. She has graduated and gone to Harvard for a postdoctoral fellowship, and the other students are still working. And I'd also like to acknowledge a longtime collaborator, John Freilinger, with whom I've done all of the cytokine work, and Dr. Cameron Cook from the University of Pennsylvania, who developed the EF5 drug and for which we did a lot of work. And of course, I've worked all this time in the Department of Microbiology and Immunology, as well as the Cancer Center at the University of Rochester, and have had longstanding grant support from the NIH. Okay, so in the last few minutes, I'd like to kind of switch gears and, is it? Yes, right. And talk about some tips for applying to graduate school. So with this, I put on my other hat. So a few years ago, I stepped back from the lab a little bit and took on the job of being Dean for Graduate Education at the Medical Center. So prior to that, I'd been very active in graduate education within the Department of Microbiology and Immunology, headed their graduate program for a few years. And so I was very, I guess, devoted to graduate education and thought the dean's job would be an interesting one, which it certainly has proven to be. A little bit more work than I had bargained for, but that's usually the case. But I would say at this time I have now looked at hundreds, if not thousands, of graduate applications. So I have quite a bit of knowledge about and tips for doing that. So I thought about this a bit and just put a few things down that I think you should be aware of in thinking about going on to graduate school. First and foremost, you want to make sure that it's what you want to do. And I don't take that lightly. It's not meant lightly. It is very important. It's a long road. It's hard work. It's difficult. Don't do it unless you're really sure you want to do it. So how do you find out that, right? Well, these are things I'm going to tell you are things you should have started doing 4 years ago or something if you haven't. But you want to do as much research as possible, the kind of research that you think you're interested in. You can't do too much. You can't get too much experience, I think. And you want to do different kinds because, you know, I knew early on I wanted to do immunology. I never wavered from that, but, you know, that's not true for everybody. So you want to try different things if you're not sure, because it might just be that it's that particular lab that you worked in that you didn't like, or it may be that you're really not that turned on by that particular area. And for whatever reasons, I think certain things appeal to certain people. I always knew I wanted to work with cells. Molecular biology is incredibly interesting and useful, but it wasn't something I wanted to do. I wanted to be able to see things, right? You know, it's everybody, each to his own. If at the time you've finished your undergraduate education and you're not sure about whether you want to go to graduate school, don't do that just because that seems like the next thing to do. If you really think you want to do research but you're not sure, I would advise getting a job and working as a tech for a year or two. You'll learn a great deal. You will find out whether or not it's what you want to do. And personally, I look very favorably on students who have worked for a couple years. And the reason for that is because I know they've decided they really want to do this. And they're a little older. That doesn't hurt. Maturity goes a ways. So I think it's a good thing. Obviously, you want to talk to other students, graduate students who are doing it. Find out as much as you can. There are also a lot of what are called post-baccalaureate programs. I run one at the University of Rochester that are open to underrepresented minorities and people with disabilities. They provide 1 and sometimes 2 years of training to people who are qualified, who are interested in getting into PhD programs. So if you're interested in that, this is another way, although I would say these post-baccalaureate programs are graded, if you will, on the percentage of students that go on to graduate school. So for these programs, they want to take people who are pretty sure about going on, but it is a good way to try out and get more experience. But again, make sure it's what you want to do. Okay. So again, one important part of doing the graduate applications are the GRE exam. Do I like the GREs? Do I think they're important? It doesn't matter what I think, right? It matters that the schools use them, and so you have to do as well as possible. At the University of Rochester, we try to look at the whole package. The GRE is just one thing that we look at. But for some schools, they use this as a cutoff. And if you don't have a certain level, they don't look further at your application. So doing as well as possible on these is very important. I find that a lot of my post-bac students have a lot of test taking anxieties. You need to figure out a way to get around that. You need to know the material as well as possible because these are timed exams. And you have to not know things, you have to really know them, right? And there's a difference if you're going to do well on these. You want to take these early because it's going to determine to some extent the level of school that you can apply to. So take them early. I don't say take them often. I find that people, unless they're willing to do a lot of work, don't change their scores very much. I've had students who've taken them 3 times. They've gotten almost exactly the same score every time. Obviously, if you don't do well and you have time, you need to prepare more and try to take them again. But it's hard to raise your scores has been my, although I think it's possible. But. So I think this is the other major decision, is deciding what schools you're going to apply to. And that's just a lot of hard work to figure out. You have to do a lot of homework on it, look at as many websites as possible, trying to identify, obviously, the more you know about what you think you want to do, the easier that is because you can hone in on the programs. You should get suggestions, right? You've all been presumably working for people who are doing research. Ask as many possible about places that they think would be good for you, particularly if they know you well, right? Because that gives them a better idea. I would say don't restrict yourself geographically. You know, when I started to go to graduate school, my mother wanted me to go to the University of Kansas. Not surprisingly, she thought California was a big evil in the West and New York was a big evil in the East, and I promptly went to both places. And it was the best thing I ever did, right? You know, you have to spread out, you know? It's a big country. There are, so don't restrict yourself, right? Look all over the country. And again, you want to apply to the very best schools for which you're qualified, but you also want to apply to a range of schools. I would say that in the past few years, it's gotten more difficult to get into graduate school than ever before. Certainly, if we're any example at the University of Rochester, we're taking fewer students, we have more applications, the bar is higher. So you want to apply to a range but also hopefully get into the best possible school. Okay. Okay, the personal statement, very important part of your application because it is a picture of you, right? And you want it to be that. You want it to be a reflection of your values, your beliefs, particularly your experiences. So your research experience should be a major component of this. You want to write this in a very logical manner. Figure out some way that works. Often that's chronologically, although it doesn't have to be, but something that will be easy to follow. And you want to show your enthusiasm for science. Everybody's looking for, you know, they don't want to feel that you're doing this because you don't know what else to do or something, right? Again, it goes back to this making sure it's what you want to do. You want to tailor this personal statement. If you apply to 12 schools, you should have 12 different personal statements that mention somewhere in that either faculty members whose research you're particularly interested in or particular reason why you're interested in that school. Because I have seen admissions committees say many times, I don't see any reason why they're applying here. They don't seem to show any particular interest in our school. So. And so write that as carefully as possible, get help doing it, you know, have a lot of people read your personal statement. Obviously, in the end, you have to write what you feel comfortable with, but get advice on that. Make sure that it's understandable and that realize that you're going to have to do, like most things that one writes, you have to do multiple revisions to make it work. And then proof it very carefully. I and I think many other people have a little tolerance for typos and multiple misspellings. It's not a good idea. Right. Okay. And finally, I think this is the very important component, is the letters of recommendation, because I know that strong letters of recommendation can take an application from something that's just very mediocre and like everybody else's to something that really stands out. So you always ask your research mentor or mentors, if you have more of those, because if you didn't write those down as a letter of recommendation, that's somebody that if the admissions committee doesn't call and ask them how they were, I will do that as dean. So it's very important, because what we're buying as graduate students is research, right? We want to know that's what you want to do and that you have some skills in that. And it's really your research mentors that are in the position to address that. Obviously, sometimes you choose professors that you didn't do research well. They need to be people that know you well. So this is, again, examples of things where you need to have started long before you're a senior undergraduate to lay the foundations for having people know you and making good impressions in class. And once you've selected that list of schools, you want to give a very clear list that has the application deadlines, because if they're getting asked by you to write a letter of recommendation, they're probably getting asked by many other students as well. I know that because of I write a lot for my prep students, and having, knowing exactly when the deadlines are is very important. So finally, you want to track all of your applications, make sure you get things submitted on time, follow up with those applications if things, you don't get a confirmation of those being finished. And really pay attention. Make sure if you have questions, you can follow up with those and contact people at the school. So I've not put up obvious things here like your GPA. Again, one of those things that you need to start on when you're a freshman in college, not when you're a senior. But that's going to follow you everywhere. And if your GPA is low, then you're going to have to correct that in some way, either through a post-bac program or getting a lot of additional experience by working as a tech and also taking some classes while you're doing that. So those are all, and again, things that if there's reasons for low grades and things, you may want to mention those in your personal statement. You want to be honest with people, lay it out. Some people screwed around for their first 2 years, but then if they've improved, that's usually looked upon favorably.